Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Hello, I'm Blue Plunkett. Welcome to episode 1 of a series of podcasts we are doing on biosecurity and bioprotection. Today we have Dr. Andrew Krylacek, leader of the molecular sensing team at Plant and Food Research. Andrew is involved with developing world-leading biosensing technology using insect models. So before we get too far into that, hello Andrew, welcome and thanks for joining us. So I guess a good place to start might be, how did you end up in this sort of bioprotection, biosecurity research?
Hi Blue. I sort of took a tiki tour to get to here, I think. Some scientists sort of spend all their life on one particular thing. I started off working on human growth hormones, isolating them from pituitary glands as an honours student. Then went to Australia, did a PhD in proteins involved in DNA replication in bacteria. And then after that, went to Spain and worked on a protein involved in cell division in bacteria. And then I came back to New Zealand and shifted into sort of the plant and insect area, and am now working on insect odorant receptors and leading the molecular sensing team here at PFR. And I guess the key thing has been a focus on proteins. They're sort of the workhorses in the cell, and of just how they work and how we might be able to use them.
Right, very cool. So you did sort of try your hand at a few various things before you ended up in this area. So you've got all this research capability which can help New Zealand's bioprotection. But so what is bioprotection, and what is New Zealand trying to protect that this research is necessary?
Okay, so bioprotection or biosecurity is really trying to prevent the entry of diseases and pests into New Zealand, and that's particularly important for our primary industries, agriculture, horticulture, aquaculture, the forestry industry. We don't want these bugs in here because they'll destroy our orchards or our forests. The other thing is that if we do have them, and we do have some of them now, that can affect our market access. So other countries won't want to buy things from us if they know that we've got the pests. And the other thing is also New Zealand's sort of native flora and fauna. You can take myrtle rust, for example, this latest this evasive fungus which is here, which could destroy our pohutukawas, a national symbol, Christmas tree. So there's a lot on the line in terms of protecting our industries and our country in general. And so we need to find new technologies and new ways to actually prevent these things coming in at the border or pre-border. Otherwise, we have to go into emergency mode and try and knock them out when they're here.
Right, so you're talking, so it's more of a reactive or a proactive type situation?
Proactive and reactive. So, let's try and stop the bugs before they get in, and if they do get in, let's eliminate them before it gets out of hand.
Right, yeah. So you can easily understand the importance of protecting the industry that's here from diseases and pests that might not be here yet.
Yep.
So traditionally, for the current level of technology was available, how was this done in the past? Was it much of a problem before, or is there sort of means that have been used before the science really came into it that worked, or has it always been a problem?
I think it's always been a problem. I think in the past we haven't really cared, but now that we've got such great industries, to be truthful, the technologies that we're working on, probably all going well, won't be available for at least another 5 years. But the only way that we can do at the moment is to randomly screen. I know MPI at the airport and ports will look at paperwork. They'll look to see what's coming in. If there's something a bit strange in the paperwork, they may say, let's go look at that container and open it up. But I think they only really look at about 5% or less of containers. And sometimes that's just opening the door, right? It's not taking anything out to find things. So what I'm hoping is that the technologies we're developing will give an MPI officer, for example, an advantage to be able to scan inanimate objects like cars or furniture coming in for pests like brown marmorated stink bug, which is this horrible thing which could destroy our horticulture industry. To be able to do it in real time and make a diagnostic decision right there and then, rather than just having to randomly open containers. One of the things we're proposing is a device that can go into a container as it's being shipped, and it can detect the aroma of an insect pest, and send a warning when it arrives into the port, and the MPI officer will know, okay, let's go and check out that particular container because it looks like it may have a pest in there.
Very cool stuff. So this type of aroma detecting device, so this leads into probably closer to the research that you do.
Yep.
How do you end up with something like that? Is this something you get from the natural world, or is this developed in the lab, or how are you detecting the aroma?
Well, okay, so there's a number of ways people are trying to do it, and they're having great success. So a lot of people are using nanomaterials to develop sensors. They might put chemical groups on that nanomaterial, which enables them to bind a particular odorant or volatile chemical and get a response. That's still got a ways to go. It's getting better and better, but it's not as efficient as it could be. And we've decided to take the alternate approach and let's use what nature's provided us. So in insects, 2 million years of evolution has produced smell receptors, which have a greater, many orders of sensitivity than current physical techniques. And insects, they're actually quite clever. You may not think so when you kill them, but if you look at a fly, it only has 24 receptors, odorant receptors or smell receptors, but it can detect hundreds of compounds, literally hundreds of them. So what we're trying to do is basically replicate the fly, but throw away the fly and just keep the protein molecules that are involved in smelling, okay? Putting them onto a biosensor and then interpreting the signal from, say, 24 receptors to say, oh, okay, I can detect Queensland fruit fly in this fruit. Maybe it's got eggs or larvae in there. Or I can detect the aroma of brown marmorated stink bug in this car seat in this imported car. Let's go and check it in further and see if there's anything in there. Or let's just spray that to be absolutely sure. So we believe that these receptors from insects are more powerful than what's out there. And we have now worked out a way to handle these things and to couple them to sensing platforms.
Very cool. That's amazing stuff, how you can utilize what's already existing in nature to do what we need to be done for testing. So, for example, you mentioned how a fly has the 24 sensors, but it can detect many more compounds than that. Is that is it a combination response? So how is it doing that?
It's a combinatorial response. So each receptor can recognize more than one compound. And some of these receptors recognise the same compound, so it's a bit hard to imagine over a radio, but usually I'll have a slide. But it's looking at the response pattern. It's like a fingerprint. And insects, their receptors, smell is really the primary sense for insects. Mm-hmm. Humans probably use vision. We use vision a lot. And they've evolved receptors for finding mates, for finding food, for working out where they can lay their eggs, to avoid predators, to avoid disease. And people have been able to show, because they can train insects to detect drug precursors or explosives. So we know these receptors are very adaptable. They can detect synthetic chemicals which a fly would never encounter in its short lifespan.
But it can still detect chemicals that it wouldn't even encounter.
Yeah, exactly.
Wow.
So that's how amazing these receptors are.
That is amazing.
Yeah.
So you've mentioned the fly as an example, but so if you're looking for like Queensland fruit fly or stink bug, is there other insects whose sensory detection capabilities are more tailored to particular compounds, or do you find that there are particular insect systems that you would use for different compounds, different volatiles?
Yeah, I think it's a bit hard to train a fly. I haven't seen it actually. I've got images of a fly on a lead.
Right.
No, the best sort of insect is probably the bee, and wasps as well have been trained to find explosives. But bees, they're actually quite intelligent animals. They have quite a social communication, in the hive and everything. And so, and we have a colleague. I've got a colleague at PFR, Flor Maas, who's trained bees to find insect pests in the field. And what she does is she uses lasers over an area, over a field, to map the movement of a swarm of bees. Okay, and see and over time see where they congregate. And the bees will buzz around a certain part of the field, and she go, okay, that's where that pest is, because I know I've trained those bees to detect that pest. Okay, so what we want to do is basically take those kind of receptors and just put it in a device and eliminate training the bees.
Right. So this device would be like a handheld, and would it just be a sound response that an MPI officer would hear if the molecule or volatile is detected?
You mean like a radioactive beep, 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 beep?
Yeah.
Maybe not. I think what we're sort of aspiring to is definitely a handheld device. It could be something which could be connected to a smartphone. So you got your smartphone, and you'd have an algorithm in there, a program, which could do could analyse the data coming in and say, look, I'm picking up a signal here. Looks like it could be brown marmorated stink bug. Why don't you keep going in that direction and see what happens if it gets stronger? So it's quite ambitious in terms of you're basically replicating what an insect does or what a dog does, but we believe it's possible. It's not, if an insect brain can do it, we should be able to do it, right?
Right. Yeah.
The smartphones are clever enough as they are now, so it's all about developing the correct kind of algorithm to interpret sort of the global response of an array of these receptors. Yeah.
Right, and an array sounds very interesting, how you could have the same array you could use across the board for all types of things you're trying to pick up at New Zealand's border, for example.
Yeah, exactly. So the vision is something, and we've just got some funding through a consortium called Better Border Biosecurity, which is a bunch of Crown Research Institutes like PFR and MPI. And the idea is to develop for them a tunable device. So let's get a good bunch of receptors on our sensor, on our biosensor, and be able to detect a range of different fingerprint aromas. Okay, so we're focusing on Queensland fruit fly infestation in fruit. So finding fruit which has got eggs or larvae.
Is it a single pheromone or odor that the Queensland or any given pest insect gives off, or can you sort of pick between a range, or is there a particular one that you find for each particular species that you need to detect?
It can be all of the above.
Okay.
I mean, for example, we found that there are at least, I think it's 5 particular volatile chemicals which indicate the presence of eggs and larvae from Q fly in fruit, and they're consistently found in different types of fruit tested. Okay, with brown marmorated stink bug, they've got particular pheromones which we might be able to pick up, but more than likely we'll be picking up a range of volatile compounds which together say this is Queensland fruit fly, or this is brown marmorated stink bug, or this could be myrtle rust, or whatever. Yeah. Right.
So one thing I was wanting to ask about was potential exciting applications of this kind of sensor work for humans, if there's any scope for that.
Yeah. So the thing with this kind of technology is that the scope is massive. Okay. If you look at what sensors there are out now, no one's fully knocked over the smell of the sense of smell or taste. You know, we've got sight, we've got hearing, we've got touch. That's all been automated, and there's huge opportunities in medical diagnostics. So we know that on the human breath, there's at least, I think, 2,000 different compounds on the human breath.
Wow.
And every day these are being correlated with disease states. So the idea here is early diagnosis saves lives. So maybe just by going routinely to your GP, he might have a device, hopefully with insect receptors in it, our device. You'd blow into it, and you'd have an algorithm on a computer or a smartphone saying, we're picking up some biomarkers here which indicate maybe some kind of cancer or early cancer development.
Wow.
Or there's some other sickness that you've got, or diabetes, ketones on the breath. So it could be a really useful first stage diagnostic tool for doctors, and they go, well, okay, this looks like there could be problem here. Let's go send you down to one of the labs and get tested properly.
That is very cool as a sort of humanitarian diagnostic tool.
Yeah.
Yeah, it's very awesome. So as far as the biosecurity aspect of this type of technology, what do you envision as its maturity, like as the future position of screening at the border? How would this look for an MPI officer, for example?
For example, I would be saying that every MPI officer would be having this this tool on their belt, right? And when they're going around at the airport talking to tourists coming in or scanning baggage.
Because at the moment we people see dogs, right? There's sniffer
Yep.
dogs, and I guess that's how the closest we might encounter some sort of biosensor setup.
Yeah.
So we'd see the same MPI officer, but not with the sniffer dog, but with some other device.
No, I wouldn't say that. I think you're always going to have the dog.
Okay.
I think I don't know if you've been to. I've been to one airport where an MPI officer came around with an Alsatian. I think it was. I could be wrong. Sorry, but just having that presence, people respect the dog. People find the dog cute, and it's a great way to bridge the gap between the officer and the public. So you'll always have the dog, I'm sure of it.
Right.
But this will be used as another form of screening, just to augment what the dog's doing. So I'd see an MPI officer would have that on hand to work with. I'd see it possibly in a mailroom coming in. You might have sensors in the mailroom smelling odours coming off the mail to see what if there's anything which indicates food or the presence of bugs or whatever in there. I would actually see it further because we have sort of this vision now at PFR of sort of a smart and connected future where you've got sensors through the supply chain. So that's where you've got orchards leading to pack houses, storage in the containers, all the way to the consumer. So
multi-level checks as it goes through the production pipeline.
Yeah, and you could be looking for pests coming through. You could be, this is after an incursion, for example, you could have them out in the field, in the orchards, like an early warning system. Sensors looking for plant stress, for example, which indicates, there's something wrong out there, let's go and check it out. So there's a huge aspect in terms of biosecurity, but it's also about crop protection as well.
Yeah, that is very cool how it's there's a specific target that you want to design it for, but the way it applies generally like that to even humans is quite impressive, quite amazing.
Yeah, we're hoping so. I mean, it's we've still got a ways to go, but we're now building a prototype, so that's awesome. That's very exciting.
Yeah, awesome. Yeah. Very cool. Well, thank you very much for having some time today to come and chat with us about your research and your work.
Okay, no worries. It's been a pleasure. Thank you.
Also, thanks to our listeners for joining us. Next time, we will be speaking with Dr. Jo Bowen about her research on host microbe interactions, specifically how fungal pathogens infect plants. If you have any questions or topics you would like to hear about, please email us at sigest@plantandfood.co.nz or see our podcast page for the link.